Welcome to my channel again, and today I come with another video, and that is the formation of the ES complex. Okay, so ES complex, that means you all know that the enzyme substrate complex. Okay, so we know that enzyme has the active site and the substrate actually bind to the active site and to form the ES complex. But there are three types of situation maybe arise, okay, in the formation of ES complex, and today we learn about this. Yeah. So first of all, you see here the single substrate reaction. Single substrate reactions means only one enzyme and one substrate can bind here. This is called the single substrate reaction. One enzyme, this is enzyme, you just imagine this is enzyme and this is the substrate. So only one substrate can bind with one enzyme. Here, this is a single substrate reaction. The second one is the bisubstrate reaction. Here, more than single substrate. That means enzyme is one. Suppose there is one active site and there is another active site. Okay. So enzyme, more than two enzymes, more than two substrates can bind with one enzyme. This is the bisubstrate reaction. Under this bisubstrate reaction, there are the two kinds we can see. One is the single displacement, another is the double displacement. In case of single displacement, there the two substrates bind with one enzyme, one after other. That means here the formation of the complex is ES1S2. This form, okay. That means two substrates binds with one enzyme simultaneously, and so the complex that is formed here known as the ternary complex. Here, this is called the ternary complex. And in the second situation where the double displacement process, we see here each of the substrate binds singly with one enzyme in ordered sequence. And that means, suppose first of all this is another substrate and this is another substrate here. And E and S1, when S1 binds with the E to form the ES1, and that displaces to form E and P1. And this E also now free for binding with the ES2 and to form the ES2. That again release to form enzyme and the product. Here what we learned today, that is the single displacement process and the double displacement process. Single displacement process, there one after other, that is one after another, they can bind with one single enzyme. Clear? And here how the products are formed, ES1, ES2, that changes to EP1, P2, and that changes to EP2 plus P1. Okay, first product is formed here, you see that. But in case of EP2, that again breaks to form P2 and E. Now E is free now. Clear? So here the ternary complex is formed, but here the two substrates cannot bind with single enzyme simultaneously. So there the binary complex is produced. Okay. So these are the process for the formation of ES complex, and these process are followed by the substrate and enzyme to form the ES complex. Now we learn about the models of the formation of ES complex. What are the models? The two models, that is Fischer, scientist Fischer, according to his opinion, the model is known as the lock and key model or the template model. And according to Koshland, the model is known as the induced fit model. Okay? According to this model, the enzyme already or previously structured for binding particular substrate. Okay, you see here the just arbitrary drawing. The enzyme, this is if this is enzyme and this is the substrate. So you see that enzyme is already structured for this particular substrate. That only this substrate can fit it with this type type of enzyme. Clear? You see that. But here one demerit is that this type of model cannot explain the allosteric modulation. And here I mentioned it that allosteric modulation I explained in my previous video. You just check out that one. Okay. What is the allosteric modulation? Next one, the induced fit model. Here the enzyme structure, enzyme actually is not rigid. According to this model, enzyme is a rigid structure, but according to this model, enzyme is not a rigid structure and it is not previously structured for binding a particular substrate. Here actually the substrate, it has a particular conformational structure. When this substrate contact with this type of enzyme, as usually the enzyme is not a rigid structure, so it can changes according to this type of structural formation of the substrate and form this type of ES complex. Clear? So this is the induced fit model, and according to the scientists, this model is appreciated by the scientists. But this model in first situation this model was appreciated, but when the allosteric modulation is discovered, this model is there is demerits is created for this opinion. Okay, so this is the formation of ES complex, and these are the model of formation of ES complex. Now we learn about the factors. Which factors are very important for the formation of the ES complex, and these factors we learned here. Now, okay, then we learn about the factors that are very important, that are very responsible for the formation of the ES complex. One factor, that is enzyme, second factor is the substrate, third one is the pH, and fourth one is the temperature. Okay. So first one, the enzyme concentration. When the enzyme concentration is slowly increased, the enzyme activity or the enzyme action, the velocity of the enzyme action also increased slowly. But after some time, when all the enzymes that are occupied by the substrate, then the velocity becomes stopped. But continuously it will increase until the any enzyme remain free or no bound with the any substrate. Okay, in the medium. So here you see that a linear graph is produced. Okay, this one is the enzyme concentration related to enzyme activity graph, and you see this is the linear graph. So here what we learned that E is proportional to the velocity of the enzyme action. Second one is the substrate. It is same like the enzyme concentration. When the substrate concentration is very low in the medium, but the enzyme concentration is high, then after some times the formation of the ES complex becomes stopped, or the velocity of the action that becomes stopped. Clear? But when the substrate concentration again we add within the medium, then the velocity rises. So here the substrate concentration is very important, just like that the enzyme concentration. But the same thing is happened here also, that when the all substrates that are totally occupied the remaining all the enzymes, okay, so there is no free substrates, but you can see that enzyme is free, but enzyme is free, but substrate is totally occupied by the enzyme. So in that situation, the activity or the action of the formation of the ES complex that remains stopped. Okay, so very important, enzyme concentration and the substrate concentration, because within on the substrate concentration and the enzyme concentration, the velocity that totally depends. Another important character, or another important character not, that is the factor. This is the pH. What is the pH factor? The optimum pH is very important for the enzyme activity. That means when the pH is optimum, then the enzyme activity is maximum. But below the optimum pH or the above the optimum pH, that the enzyme activity. Sorry, one second. Sorry for the disturbance. So here what we learned that optimum pH. The optimum pH is very important. When you see that this is the graph, it is a hyperbolic, you can say, or the bell-shaped graph. So here the 7 pH, when the pH is 7, the maximum velocity you can see, and this is the 100%. Okay, and enzyme activity here here rises maximum. But below the pH 7, that is 0, or above the pH 14, you can see the velocity or the enzyme activity become decreased. Clear? Next one is the temperature. Like that pH, optimum temperature is very important. That is generally 37 degrees Celsius is very important, because in that optimum temperature the enzyme activity is maximum. But below this optimum temperature, the enzyme activity, or above this optimum temperature, the activity of the enzyme action that becomes stopped. Another two important factors that are I don't write here, I just explain you. The last one is not last one, the reaction products. Okay, the reaction product means when the enzyme complex is formed, ES complex, then it not stable, it is not stable, it immediately changes to E plus P. So product is very important. When the product, the amount of product is high, then the enzyme activity becomes decreased. Okay, it is very important, the amount of product that remain within the medium. Okay, so when the product is present within the medium, then enzyme activity that becomes stopped. Okay, and again another is the inhibitor. Inhibitor is another important factor, that is very important. When inhibitor is present within the medium, then it binds to the E to form the EI complex. So naturally you can see, if EI complex is formed here, that means ES complex cannot be formed. Okay, and now here I tell you that enzyme inhibition I explained in my previous video. You just check out how this enzyme inhibition is occurred, and it is affected, or it affects on the Vmax and Km. So you just check out. Okay, so it is the EI complex, and as usually if EI complex is formed within the medium, then ES complex cannot form in a regular basis within the medium. So here inhibitor is another important factors. Okay, so what we learned here, that if the product accumulates within the medium, that the enzyme activity becomes decreased, and if inhibitor is present within the medium, then ES complex formation also decreased. Okay, so these are the factors we learned here. Here we learned that how the ES complex can be formed, and all the factors that are very important or responsible for the formation of the ES complex, or for the activity of the formation of ES complex. Clear? So here I think you can understand it clearly, and I totally explained the total enzymes, all the topics that related to the enzyme, and this is the main important part of the biochemistry. I explained in my different videos, and you just check all the videos. And if you like these videos, obviously give one like for these videos, and share and subscribe my channel. Okay, that's why I can also provide you another knowledges or about any topic that related to the biological science. Clear? Okay, thank you.